Hello everyone, I myself Dr. Priyanka Shanoyan from Department of Biotechnology. So today I am going to speak about cloning and expression of secondary metabolite genes. So foremost importantly, if we have to concentrate on certain secondary metabolite genes, we have to concentrate on two important aspects, that is with respect to medicinal plants, because these medicinal plants are it consists of lots of secondary metabolites inside it. So if we have to concentrate that particular secondary metabolite, foremost important requirement is gene of interest. So if we have to concentrate this gene of interest, we have to isolate this specific, we have to identify as well as isolate this specific gene of interest in an array, in a pool of DNA, that is in a genome. So if we have to isolate that, foremost important targeted pathways has to be concentrated. So foremost important pathways in the medicinal plants are flavonoid pathway, all these are secondary metabolites, and terpenoid pathway, alkaloid pathway. And in these pathways, if we have to take, there will be a foremost important key enzymes. If we target that key enzyme, why it is called as key enzyme? Because it's a rate limiting pathway, particular step pathway genes. So if we have to target this, so then it will be very beneficial for us, because it will be coding for certain different secondary metabolite as an end product. So why the secondary metabolite? Usually in the plant system, there will be two different types of metabolites that has been synthesized. One is primary metabolite and the other would be the secondary metabolites. These primary metabolites are required foremost importantly for the growth and survival value of the plant system, whereas in the case of secondary metabolites, these are synthesized in very lesser concentration, but these secondary metabolites has a higher value, that is it will be significantly important pharmaceutical product, so which has a commercial significance. So hence these type of genes can be concentrated mainly by studying the three foremost important pathways, and there will be multiple different pathways that are correlated. So if we have to concentrate and isolate the specific gene of interest, and mainly cloning can be done. Cloning, how it can be done? Foremost importantly, we have to concentrate on a important plasmid for which you are going to add a specific gene of interest. What is cloning? Multiple synthesis of the genes can be retrieved mainly by taking the important vector system. What are these plasmids? Plasmids are extra chromosomal self-replicating double-stranded DNA molecules. So these plasmids now has to be which are commercially available. These plasmids can be isolated, and these plasmids can be retrieved, and these plasmids holds this has a capacity to hold a significant genes. So which genes we are concentrating here? That is a important secondary metabolite genes. So what is the important activity that is foremost important necessary in order to clone? First, there will be a different types of restriction enzymes, and there will be specific origin of replication, and there will be a specific important gene of interest where we can add into the specific plasmids. So right now our basic job is to linearize these particular plasmids. So there will be specific restriction site wherein a particular restriction enzymes cut this particular plasmid in a significant site, and this will get linearized. And after this, you can add a specific gene of interest whichever we are in need. For example, if it is a flavonoid pathway, for example, if it is a terpenoid pathway, and there will be an alkaloid pathway. In this foremost important in the terpenoid pathway, foremost important pathway genes that can be phosphomevalonate kinase, and all these are certain key enzymes. So how we can target this particular key enzyme? There are various in silico analysis tool right now. So one of the important tool which we can isolate wherein we can target this specific PMK gene is through anti-smash analysis. This is nothing but antibiotic and secondary metabolite analysis shell. This is a foremost upcoming in the research right now. It's an in silico tool foremost importantly required in order to identify some of the important secondary metabolite genes, and this will provide an in silico data for providing an important genes which are there, and it will give the complete sequences. So when we get the complete sequence of this particular gene, foremost most importantly required, so that particular genes can be targeted, and in that we can design the specific primers. So as a result of which we can retrieve the specific gene of interest which is required, and you can combine that, we can add that to the specific plasmid. Once this plasmid is combined, mainly DNA ligases, these are called as molecular gums. So this can be attached to this. So this, what this DNA ligase helps in, aid in, this will specifically act as a gum system. So this will bind your specific target gene with this plasmid. Now these plasmids are called as recombinant plasmids. Different types of recombinant plasmids can be created, and nowadays we will get multiple different commercially available plasmids. So for example, PET, PRSF vectors, different availability of plasmids are there synthetically. So right now we can isolate that, we can take that, and we can specifically add our own gene of interest into the specific plasmid, and we can create this recombinant plasmid. Once this recombinant plasmids has been created, later you have to add this plasmid into the particular system, that is inside the bacterial system. So inside the bacterial system there are various different methodologies, that is gene delivery techniques, wherein you can adapt. Foremost important gene transfer technique, that is adapted through transformation, conjugation, transduction, wherein you can add a specific gene of interest provided by a plasmid inside the particular cell. And foremost importantly we can adapt is by through the heat shock. What this do, there will be a permeability, there will be a creation of a particular holes wherein your recombinant plasmids gets inside the bacterial system. And this bacteria, suppose if it is E. coli, right now it has a capacity in order to multiply within 20 minutes. So these plasmids gets replicated within 20 minutes due to its doubling time, as a result of which you will get a multiple copies of the bacterial system. This is how you can easily target, you can easily incorporate the particular plasmids inside the particular bacterial system. So there will be a clonal proliferation of this bacterial cells. So hence multiple copies of this bacteria definitely contain the gene of interest provided by the plasmid system. Later, due to the expression, your secondary metabolite important drug molecules can be retrieved. Foremost importantly, how we can concentrate some of the important medicinal plants, because it has a pharmacognostic values. So how? Because it has a it has a specific importance which has been provided through the synthesis of this multiple compounds like alkaloid, terpenoid, flavonoid, different types of secondary metabolite compounds has been synthesized inside the medicinal plants. And foremost importantly, we can make a note that inside the medicinal plants there will be wide variety of endophytes which resides inside the medicinal plants. What are these endophytes? Endophytes are the one, endo means inside, phytes means plants. Inside the medicinal plants there will be multiple diversified fungal system, that is fungi, and there can be endobacteria, endofungi, endophytic fungi. So these also associates with these medicinal plants in order to synthesize multiple variety of secondary metabolites. So how this can incorporate because of correlation, because of co-mutualism, because one will be beneficial, there will be a symbiotic association between these two, between the medicinal plants as well as the endophytic system. So as a result of which even they both can synthesize certain different types of drug molecules which has a commercial significance, which has a different activity like different types of anti-malarial drug, anti-cancerous drug. The best example is Taxol, which can be retrieved. And if we want these concentration of secondary metabolites in much higher concentration, so we can adapt these types of strategies wherein this can lead to the multiple synthesis of important drug molecules or which has a commercially significant molecules. Thank you.